work was done as part of our early EAP program with TwoBind, another longtime collaborator, and is available currently as a preprint. In order to successfully develop vaccines, we need stable antigen proteins in order to study in the lab. One of the challenges of creating an HIV vaccine is that the native envelope protein has not made a good target for vaccine development. An engineered derivative, BG505-SOSIP, has been developed but requires further characterization for stability and vaccine development. TwoBind has a screen of FDA-approved buffers for biologics development and wanted to know how these buffers affect the long-term stability of the engineered HIV envelope protein. To do this, they wanted to do long-term stability studies. However, by definition, these studies take time, and time from starting in the lab to final patient development is crucial in developing these vaccines. So they wanted to find something that would save them time in the lab. Additionally, they also had a lot of buffers to screen and didn't want to use too much material for each step along this way. Here we can see some highlighted data from the full screen. Buffer 9 in black offer the highest melting temperature, which implies the most stability. On the other hand, buffer 70 in blue has the lowest TM, implying the least stability. However, as we can see, buffer 57 in red also has a fairly high melting temperature, but also was determined to have the lowest PDI of any of the buffers. When we look at the relative frequency versus hydrodynamic radius graph, we can see how buffer 57 does not have any higher order aggregates forming, but buffer 9 in black does. Compare this also with the least stable protein, buffer 70 in blue, which also has a high PDI and shows that large aggregate molecules are forming. Compare this, because of the insights gained from DLS, they went on to further characterize buffer 57 with accelerated stress studies and antibody interaction measurements, not shown here. Also important to note is that because the nano-DSF, turbidity, and DLS measurements were all taken as part of the same measurement run, doing all these experiments was incredibly efficient. Not including the incubation times for the accelerated stress studies, the full workflow from 96 condition screen to stress studies with the final candidate required only about 5 days of hands-on time.